Hello everyone, my name is Lam Bang Hao. I am a PhD student at the University of Science and Technology. I am currently working at the Korea Research Institute of Chemical Technology. On behalf of my co-authors, I am pleased to present you my IRPAC Macro 2020 Plus presentation titled Functionalized Bioorganic Nanoparticles as Strong Adhesives for Biological Tissues. So in surgery, wound closure usually achieved through the use of staple and sutures. However, these methods are very damaging to healthy tissues and they are not always applied to the inner body parts. So another option is the use of polymeric hydrogel adhesives, like the bandage you usually use at home. However, the critical limitation is that they glue very weakly underwater because water dissolutes polymer. And even though scientists try to develop some underwater adhesive like the one inspired from muscle or tunicate, their adhesion require not biocompatible conditions like basic pH or the use of cytotoxic cross-linkers. That's why people have to develop another class of adhesive that can both glue underwater and biocompatible at the same time. In this study, we developed a novel type of tissue adhesives based on three sources of inspiration. The first one is the discovery that inorganic nanoparticles can act as tissue adhesives, and this is published in Nature in 2014, and the use of inorganic nanoparticles is convenient because its suspension is not very highly viscous. However, inorganic nanoparticles don't have a specific functional groups on their surface, so they adhere weakly to tissue. And secondly, the use of inorganic nanoparticles raise some health concerns. So that's why we take the second inspiration. We decide to choose a natural source, which is chitin. Because chitin can be modified into nanomaterial as well. And chitin nanomaterial is highly crystalline, so it can create a strong cohesive force that prevents it from being dissolved in water. And the third inspiration is we try to modify the chitin nanomaterial surface with functional groups so it can perform strong adhesion underwater. And we modify chitin surface based on observation that in living system, which is made mostly of water, there's strong interaction between biomolecules, and which is usually mediated by guanidinium moiety shown here in blue color. This moiety is special in a way that even though it has positive charge, it also has some hydrophobic characteristic. So besides ionic interactions or hydrogen bond, it can also perform hydrophobic interaction, and we can combine all these characteristics together to create strong tissue adhesive underwater, and it's still compatible compared to other type of tissue adhesives. Show here is a scheme of how we got guanidinium chitin tissue adhesive. First one, we transform chitin into nanomaterial through treatment with acids and then base, and then we perform direct modification on the chitin nanomaterial surface through reaction with cyanamide under suitable catalyst. And we confirm the surface modification through the C13 NMR, which show an additional peaks for the central carbon of the guanidinium moiety. And the SEM image also confirmed the nano size of our glue. And finally, the glue dispersed very well in water. The reason is that guanidinium have a very weak acidity. Its pKa is larger than 12.5, so pH7 is kind of acidic to this functional group. Therefore, it creates positive charge on this functional group, and this positive charge is help stabilize the suspension. So after confirming the chemical structure and the stability, we test the adhesion strength of guanidinium chitin glue on software surfaces. Shown in the first picture is the two pieces of gelatin hydrogels, which is glued by the guanidinium chitin adhesive. and it can sustain a 2-gram weight. The second picture shows that 2 pieces of porcine skin can also be glued as well. And when we measure the adhesion strength, we observe that the assembly that glue with guanidinium-chitin tissue adhesive is 6 times stronger than the ones glued with the inorganic silica nanoparticles. And this inorganic nanosilica particle is the one that tests on the Nature paper. So this showcases the importance of surface modification. For better illustration, you can check out the video attached here. As you can see, the glue can be simply applied by just dropping and then pressing with fingers. On the right is a commercial silica nanoparticle TM50. It cannot sustain the 200-gram weight, but on the left is our glue, which is performed very well. And when you see the failure, one is failure at the adhesions and one is failure outside of the adhesions. This slide results showcase the underwater adhesion ability of the guanidinium-chitin adhesive. So porcine skin after glue with the guanidinium-chitin adhesive was put underwater, and its adhesion strength is measured throughout 1 week, and we observed that the adhesion strength after 7 days still maintained at about 10 kilopascal, which is higher than the one exhibited by inorganic silica nanoparticle TM50 under normal conditions. And for better illustration of the application, please watch the video. Here we glue the 2 big pieces with guanidinium glue, and when we put underwater, the one without glue will form immediately, but the one that glue with guanidinium-chitin tissue adhesives only fail when we increase the stirring rate. So besides being a good tissue adhesive, the material should exhibit other higher order functions. One of them is being a good hemostatic agent, which means it can stop bleeding to prevent blood loss. So to remove the effect of fibrinate, which is a very important factor in blood clotting, we test the blood stopping ability of the guanidinium-chitin tissue adhesive on the fibrinated blood. And as you can see in the raw sample, there's no evidence of blood clotting during the test time. However, if we treat it with our materials, blood clot occurs at around 7 minutes, because when the blood solidifies, there's a crossover point of the storage modulus and the loss modulus. And when you observe in the picture, you can clearly see blood clot from around the plate, while in the raw sample, there's no sign of blood clots. And when we lift the plate over, there's a lot of blood liquid that lost over the plate. So another interesting function is antibacterial, which can prevent the wound from being infected. So we test the antibacterial of guanidinium-chitin tissue adhesive, and we observe that our material possesses very strong antibacterial comparable to the positive control using copper sulfate. However, the precursor used to synthesize our materials, which show here in orange, show weaker antibacterial effect. It only inhibits bacterial growth. So we conclude that the interaction between these two materials is different because they have different surface functional groups, and the guanidinium moiety binds to the bacterial stronger than the amino moiety, so the binding can disrupt the bacterial membrane function and lead to cell death. And as a confirmation, you can see here the wells filled with the guanidinium nanochitin had a larger inhibition zone compared with the precursor, and its effect is comparable to the positive control. So in this part, we try to understand why the guanidinium-chitin can bind strongly to tissue. Because the material bears a positively charged functional groups here, we try to break down the amino acid composition of gelatin and porcine skin, and we observe that Among various interactions, two of the most common and possible interactions is among the hydrogen bond between the guanidinium and the hydroxyl groups, or ionic interaction or bond bridge between the guanidinium and negatively charged. And I should emphasize that these are just very simple interactions among many others. So to better understand the adhesion mechanism, we designed a simple experiment that study the ionic interaction caused by guanidinium moiety. So shown here in this configuration, one surface is coated with the guanidinium moiety representing our adhesive, and the other surface is coated with the carboxylate groups represent the composition present in the hydrogels and porcine skin. And then we brought the two surfaces close together until they come to very close approach, and then we pull them apart. And as you know, when we pull them apart, it requires force, and this force is proportional to the adhesion energies that hold the two surfaces together that caused by the two functional groups. And when we measure the adhesion strength, we observe that on the PGA, the one that bear wholly the negatively charged, the guanidinium exhibits a very strong adhesion energy compared to the chitosan with by only the primary amino groups. And when we change to gelatin, the adhesion is upgraded a little bit higher. So this demonstrates that besides only ionic and hydrogen interaction, there's other more complex interaction that can induce. I would like to conclude my presentation with this figure. We developed a novel type of tissue adhesive that combine various benefits. The first one is the convenience of use and storage by nanomaterial. The second one is strong underwater adhesions mediated by the guanidinium moiety by living system. The third one is biological benefits of chitin from nature that can accelerate wound healing. And more importantly, we discover the role of guanidinium in underwater adhesions, and we hope that this important finding can inspire the design of future adhesive so that it can glue more effectively underwater. I would like to acknowledge the financial support from the Korea Research Institute of Chemical Technology, and I would like to thank all the professors, doctors and researchers who work here and helped me a lot during this study. I also acknowledge the scholarship support from my university, the University of Science and Technology. Thank you for your interest in our work, and if you have any questions, please leave a message in the Q&A session. And further inquiry can be sent to me, the presenting author, via my email, or my corresponding author, my professor, Dr. JACS Au. Thank you very much.